San Diego Science and Technology Council is a network of regional science and technology organizations, and the purpose of the council is to enhance innovation, the innovation capability of the region. And one of the ways we seek to accomplish that is by engaging opinion leaders in science and technology policy on issues of common concern. The completion of the human genome sequence was the first step in a progression of research advances in structural and functional genomics. These advances will continue to pose significant ethical, legal, and social implications for society by challenging existing assumptions about the control of human characteristics, identity, and behavior. This evening, we have the opportunity to explore issues related to the application of advanced genetic screening technology with our distinguished guest, Dr. Edward McCabe. As a pediatrician and geneticist, Dr. McCabe is currently professor and executive chair of the UCLA Department of Pediatrics and physician-in-chief of the Mattel Children's Hospital at UCLA. He directs the Pediatric Research, Innovation, and Monitoring Experience, PRIME, program, the UCLA Child Health Research Career Development Award, the Human and Molecular Development Postdoctoral Training Program, and the UCLA Center for Society, the Individual, and Genetics. I think he came down here today just to get away from all those jobs. Dr. McCabe received his bachelor's degree from the Johns Hopkins University and both his PhD and MD degrees from the University of Southern California. Prior to his recruitment to UCLA, Dr. McCabe pursued his academic career at the Baylor College of Medicine, where he established the Baylor Mental Retardation Research Center, the Baylor Molecular Genetics Diagnostic Laboratory Postdoctoral Training Program, and the Baylor Child Health Research Center. Throughout his career, Dr. McCabe has served important roles in a number of honorary societies. In 1990, he co-founded the section of genetics of the American Academy of Pediatricians, and he served as president of the American Board of Medical Genetics from 1995 to 1996. He was elected as a member of the Institute of Medicine in 2001 and served as a member of the Human Cloning Panel of the National Academy of Sciences, and currently serves as the chair of the Secretary of Health and Human Services Advisory Committee on Genetics Testing. Currently, Dr. McCabe is president of the American College of Medical Genetics, president of the Western Society for Pediatric Pediatric Research, and a member of the American Pediatric Society Council. He has a very provocative talk for us this evening. We're very fortunate to have Dr. McCabe as well as his wife Linda here with us, and I look forward to his talk, and thank you all for coming.
Well, thank you very much for inviting me here. First, let's talk about what genomic medicine is, because it's really key to understanding how the Human Genome Project is going to impact on genetic testing and technology. It's anticipatory, not reactive. It's predictive and preventive, and it requires knowledge from genomics and derivative disciplines, and we'll talk about what they are. It will involve genetic screening of individuals and populations, so that we're going to be testing people before they get disease rather than after they have disease. And it will involve new analytical technologies and bioinformatic approaches. It's going to require a change in our culture to implement this. On the practitioner side, we're going to have to go from an acute medicine to a preventive medicine approach. And physicians aren't trained that way. They're trained to react to disease, not to anticipate and prevent disease. There's going to be less independence, I think, for individual physicians, and we'll talk about that a bit more. But if we're going to really accommodate all this technology, we're going to have to give up some of that independence and become somewhat more protocol-driven, at least on the intake side, on the testing side. For the population, it's going to involve changes in lifestyles, and we know how hard that is. From smoking, obesity, all of these issues have been difficult to implement, and we're going to talk about really implementing that more broadly in terms of lifestyle approaches. And it'll involve participation in large clinical trials to really understand the natural history of complex genetic disease and how the genes influence that natural history. It's going to require changes in medical data analysis. It's going to be driven by lab data, not knowledge, at least, of pathogenesis. You're going to find correlations. You may not understand why these genes are important in that disease until later as one does the research. But that's not new. We knew how to treat PKU before we really understood what caused PKU. So it's not that new in medicine for us to address a disease before we understand pathogenesis. PKU is a disease, a biochemical genetic disease that causes mental retardation, and it was treated with low phenylalanine before we knew it was caused by a mutation in phenylalanine hydroxylase. It has a huge amount of information. It's going to be hard for most individuals to be able to synthesize all the information, and it's going to involve, to some extent, pattern recognition as opposed to sort of simple linear pathway approach to problems. It's going to require a change in diagnostic approach. There's going to be trust that one has to place in unseen algorithms, but we do that in other areas of medicine. All the imaging that's so important to the practice of medicine today, I mean, I don't know the algorithms that go there. I know the edges keep getting sharper on the images because they change the math all the time. Again, it's not like this is the first time this will happen in medicine. Evaluation of matrices and dynamic complex systems, not linear pathways, and at least initially it's going to be less hypothesis driven. Our differential diagnosis is frequently hypothesis driven as we approach the patient. I think we're going to see an increasing reliance on protocols, but we're already seeing that happen in other areas of medicine. I think we're going to see the balance between the art and science of medicine. In medicine, we're always saying we're an art, not a science, which I argue with because many of us have spent our careers trying to make it more of a science. So I think we've seen there become a balance in art and science. We're going to shift that toward the science, at least in the predictive screening and diagnostic areas. But I think it's very important that we not shift all the way over, as we frequently do. The pendulum always swings too far. We can't lose the art of medicine because that will be essential for successful implementation of preventive and therapeutic measures. If we're going to do that implementation, if we're going to change lifestyles, if we're going to have people take medicine every day, even if it's a pill and not a lifestyle change, it's hard to get people to take medicine every day for 2 weeks straight for antibiotics. So to do that when you're not feeling a difference from it, and the difference is 50 years from now, I think is going to be very hard, and that's going to take a lot of art. That's not going to be science. What are the consequences? Well, we would hope that we might see a reduction in the burden of chronic illness. This is what we say in genetics. I hope that that'll actually happen. We'll see a decrease in the prevalence of common complex diseases. Again, this is what we predict. We'll see if it really happens. I think there's a chance that we could see an increase in health disparities between those with and without access to this technology, to genomic medicine. But then I also am optimistic that there may be some technological spin-offs in other areas of medicine, including infectious diseases, that will actually improve health disparities. We'll get some of this technology out to the third world in areas like diagnosis of infectious disease. So how have we gotten here, and what have we already learned? Well, Watson and Crick, we're going to celebrate the 50th anniversary. Magically, the human genome is going to be completed. We knew a year ago that the human genome is going to be completed on April 25th. 2003, the 50th anniversary. So we'll measure another milestone in the human genome, but there'll still be some holes in it. The genetic code of human life has been cracked, and this shows Francis Collins and Craig Venter from the public and private human genome projects at the White House. This was the publication of the 95% complete draft sequence in Nature and Medicine. The Human Genome Project was initiated in 1990, completion originally planned for 2005, but with the private efforts, it accelerated the completion, and it's now anticipated for this spring. The results involve the complete sequencing of the human genome, a new branch of science and medicine, genomics. I put these slides in because one time I gave this talk, and it was a general audience, and at the end somebody said, "What is a genome?" after I've been talking about genomic medicine. I don't think that would be an issue here with this audience, but I want to talk about the derivative disciplines. But it's also important to point out, everybody talks about completion of the Human Genome Project. The first Genome Project was actually completed in the 1980s with the sequencing of the almost 17,000 base pair circular genome in our mitochondria. We really are two genomes, the nuclear genome and the mitochondrial genome. One of the derivative disciplines is transcriptomics. If transcript is an RNA copy of a gene, then transcriptome is all of the RNA gene copies in a cell, tissue, or individual. Proteomics is a study of all proteins in a cell, tissue, or individual. Metabolomics, the metabolome is all the small molecule components of a cell, tissue, or individual that are produced by proteins of the proteome. And I met with Bernard Paulson today and heard a new one, fluxomics, where they're studying flux in complex systems. This is going to generate the complete part lists, but also the parts assembly instructions. As we all know, you need to know how to put it together. It doesn't help just to have the parts there. And so understanding all of these derivative disciplines will be required to understand the parts assembly instructions. What have we already learned? Well, one of the things is that we're not the center of the biosphere. I love this slide of Aristotle, Ptolemy, Copernicus. These were pretty bright people. Copernicus was the brightest. He first figured out that we were not the center of the universe. And he also was bright enough to figure out that he better not really make a big thing of this. Galileo, also a very bright guy, was not as smart as Copernicus. He really started telling everybody about this and making a big deal out of it, and the church wasn't real happy with him. We really share a lot of genes. This is the homeobox and shows that the way the genes are laid out, the way they're turned on during development, they're very similar in the fly and in the human, and there are lots of other examples. There's no biological basis for the concept of race. There's more genetic variation within ethnocultural groups than there is between them. Even the terminology race is erroneous conceptually, and therefore there is no biological basis for racism. Even our ethnocultural identity may be challenged by genetics and genomics. For many of us, our knowledge of our heritage is quite limited. 3-generation pedigrees. I'm a geneticist, and I was always asking people for their pedigree. And then I realized I couldn't do what I was asking my patients to do. So fortunately, I asked my mother before it was too late and found out that I wasn't who I thought I was. I always thought I was pretty Irish. My wife maintains the only thing Irish about me is my temper, because it turns out I'm 7/8 German. And I think a lot of people really know their heritage by surnames, by family lore, but if we're ever able to really determine an individual's ethnocultural group from genetic genomic technologies, we're going to be in for a lot of surprises, I would bet. Even the concept of two sexes lacks biological rigor. It denies modern genetic and social observations, and individual sex can be defined in a lot of different ways, and they all may not be synchronous in that individual. And so here are some of the modern concepts of sex and gender: sex of rearing, genital sex, chromosomal sex, genetic sex, legal sex, gender identity. And here we're already seeing impacts on families, on individuals with the genetic technology. So what we've learned is that gender, sex really is a continuum. It's not a dichotomy. There may be a conflict in sexual assignments even when using biological criteria. So an example would be a 46XX female who may have the testis determining gene called SRY on the Y chromosome may be deleted for that or have a mutation in that. 46XX male, that can happen without the Y chromosome if you have that SRY translocated to another gene, most commonly another chromosome, most commonly the X chromosome. And then you can have a 46XY SRY plus female. And this is a gene on the X chromosome called DAX1 involved in adrenal development and when duplicated causes sex reversal. So what have we already learned? Fundamental and well-accepted concepts are not supported by the lessons from modern genetics or genomics. And if we have to rethink our position in the biosphere, our ethnocultural and sexual identities, then how else is this going to influence our concepts of ourself and how we use this genetic information? I'm a geneticist, so of course, all disease has one or more genetic components. I would have said 10 years ago, I would not have been so bold 10 years ago. I would have said everything except trauma and infectious disease has a genetic basis. Now we know that response to trauma, risk-taking behavior, all of those have a genetic basis. How you heal after trauma can be influenced by genetics. The susceptibility and progression of infectious disease has genetic bases. If we accept that, there's some genetics in most disease, then there's no basis for genetic discrimination since we're all in the same risk pool. And actually, Bill Brody, who's the president of Johns Hopkins, as a loyal alum, I get their newsletters, and I just saw one last week where he had something in the Wall Street Journal, an op-ed piece, saying what many of us said, and that is that genetics, genomic medicine will drive us to a single payer, because insurance is based on discrimination. It's based on being able to discriminate who's going to be healthy and who's not going to be healthy, and then gaming the system so that you end up with money at the end of the day. And once you really can figure out that we're all at some genetic risk for some disease, it's going to be hard to game the system. For a while you'll be able to say, well, maybe cancer costs more than something else, but it's going to be hard to do that. Finally, they won't be able to make money, and then they'll turn it over to the government. So it's a fairly bold statement, but certainly something to discuss. The other reason why I've been on panels, they say, well, it's not discrimination. There's really no historical use. You have to have a history for discrimination. I point out that we passed the American with Disabilities Act, so there is a history. And it was felt that it was a problem that needed to be rectified with the legal system. I also think it's discriminatory because we discriminate against those for whom we have genetic information. But it's just that we don't know what I'm susceptible to. Because I don't have a family history of Huntington disease or cystic fibrosis. So we're only looking at those where the genes have been discovered so far. And as we study the complex genetic diseases like cancer, cardiovascular disease, diabetes, Alzheimer's disease, we're going to then learn that many of us have susceptibilities we might not have recognized. Al Carnesale, when we were meeting to put together the UCLA Center for Society of the Individual and Genetics, Al is an atomic physicist who, when he came to UCLA, he was head of the Kennedy School of Government because he got into disarmament. So he is very interested in these interdisciplinary type activities. He said, we've just come through the Manhattan Project of Biology. Let's get it right this time. The Genome Project has been doing this through the ELSI program from the very beginning, putting 3 to 5% of its money into the ethical, legal, and social issues. We're doing it now at UCLA. A lot of other universities are doing this sort of thing. The U.S. Department of Health and Human Services Secretary's Advisory Committee on Genetic Testing, and now the new Secretary's Advisory Committee on Genetics, Health, and Society is addressing these issues. And there's a lot of people, a lot of groups that are working on these sorts of issues. So now let's do some case studies. And all of these cases are real. or they've been varied a little bit. Where they've been varied from real cases, I'll tell you where the variation is. This is one that's very close to a real case. A patient who works for a small self-insured company has a positive family history for emphysema on both her mother's and her father's sides of the family. Her physician recommends that she have a number of tests performed, including one for alpha-1 antitrypsin. When the alpha-1 antitrypsin test is reported to be abnormal, he tells her that this may explain the emphysema in her family and places her at very high risk for this lung disease. Her physician reports the results of his evaluation to her insurance company as required. When you sign that form, you release the information from your medical examination to the insurance company. That's so that they can audit and be sure everything was done to you that they're paying for. When you're self-insured, the insurance company is the employer. And so this is where we have found the problems when there were small companies that were self-insured. Several days later, she's called into the office of her employer, and even though she had a file showing she was an exemplary employee, she's now fired. In the actual case, the patient had symptoms at the time of testing. So it wasn't just a genetic predisposition. She actually was beginning to have the very early stages of lung disease. And Commissioner Paul Miller from the Equal Employment Opportunity Commission argued this case under the ADA, the Americans with Disabilities Act, and it was settled in favor of the employee. What remains to be determined, and Paul's actually looking for a case that will allow him to move it the next step, and that is whether a mutation, a sequence variation, is a disability. But think about the implications of that. Then we're all covered under the ADA.
Was it settled or adjudicated?
I probably don't know enough to know the difference there. It went to, it was heard, and a decision was rendered.
An administrative decision?
I'm sorry, I don't know that well enough. Linda, do you know? Linda teaches genetics and ethics in the human genetics department. Okay. Now sorry, don't know the fine points.
It's important for its precedent-setting effect in subsequent cases.
Okay. Another one, workers for Burlington Northern Railroad. This is a real one. Gave blood sample as part of company physical examination. Portion of the sample was sent to a private laboratory for PMP22, peripheral myelin protein 22 deletion testing. This deletion is associated when it's duplicated, you get Charcot-Marie-Tooth disease. When you delete it, you get hereditary neuropathy with liability to pressure palsies, which is a slowly progressive neuropathy that may present with carpal tunnel syndrome. In fact, at the time that they began doing these testing, there was one paper in the literature, one 3-generation pedigree where there was carpal tunnel syndrome. The workers didn't know that they were having genetic testing for HNPP and did not provide consent for the testing. The union brought suit against Burlington Northern, and the testing was stopped. So what are the issues here? Actually, this case revolved around consent more than genetic testing. It was felt that the individuals had not consented to this. It was felt that there was inadequate information. In essence, this was a clinical trial that was being performed. There was some evidence to suggest that that might have been the case. Informed consent has been recommended by a number of groups for all genetic testing, but others argue, why is genetic testing different than any other medical testing? Many of us believe there needs to be a firm evidence base for any genetic test to move into the clinical arena. That's a fairly high bar because we don't use that in a lot of other areas of medicine. But at least we need to label genetic tests the way we label drugs so that we know what it will do and what it won't do, what arena has it been tested in. A genetic test may perform very differently when there's a known family history than in population-based screening. Direct-to-consumer marketing of genetic testing. 25-year-old with a very strong positive family history of breast cancer. Her mother, maternal aunt, maternal great aunt, all presented with breast cancer before age 40. She sees a direct-to-consumer ad for BRCA1 and BRCA2 testing. testing was not performed on her mother, who died in 1987, and this woman is one of 3 sisters. The cost of testing these 2 genes is $2,800 without a known mutation, because they basically have to sequence the BRCA1 and BRCA2 genes. She elects to pay this out of pocket to avoid informing her primary care physician and insurance company of testing. And many individuals do this as a way of trying to prevent genetic discrimination. But what are the implications of this? First of all, the direct-to-consumer marketing is being carried out on this. Myriad, the company that does this, decided it would go into the Denver and Atlanta markets. Linda and I were passing through the Denver airport back in October when they had begun the DTC marketing, and we, in a period of a half hour, we saw 3 runs of this ad on the airport monitor with CNN or whatever was on. It was the same genre of Nexium, the purple pill, only a little bit more, because here, and Myriad says they're trying to educate. It was more, aren't you afraid of breast cancer, and you can have this test done, ask your doctor about it. So if she doesn't disclose that, if a mutation is identified, is she going to know what interventions are available to her if she doesn't tell a health professional? Is she going to discuss with her sisters and other relatives that a mutation has been identified and what their risks are? And what about the impact on her health insurance? Again, that fine print that you signed says that if you have information that you don't disclose, then the insurance is null and void. What if no mutation is identified? And we're teaching a class at UCLA, and we presented this case to the undergrads last night, and they all recognize this. A lot of people are going to say, oh, I'm fine, I'm not going to get breast cancer. No, there's a family history. All we know is that your family doesn't have a mutation in BRCA1 or BRCA2 that we can identify. But we haven't really altered the risk for that. But people are going to be reassured. What kind of discussion is she going to have with her sisters and other relatives, and is misinformation going to be passed on? And what's the impact on her health insurance? Even if she doesn't tell them about the family history, that will impact on her health insurance when the insurance company finds out. So, there was an article back in the LA Times in August by Zittner that they were already doing direct-to-consumer genetic testing or planning to do within the next couple of months. Drug metabolizing enzymes, vitamin mix selection, personalized health and nutritional advice, like a gene that's involved in thought to place one at risk for cardiovascular disease, possibly for neural tube defects in one's offspring. Hereditary hemochromatosis, cystic fibrosis, factor V Leiden, a lot of stuff. Home genetic testing is actually being developed. The equipment is under development by several companies. I met with one of them a couple of years ago, and they said, you know, we've got the equipment. We know we'll have the equipment in a year or two. Help us figure out what the market is. So the technology is going to drive the market. And they're getting things down to a glucometer size, a size that can fit in your pocket to do genetic testing using macroarray technology. What will be the role of the healthcare provider in test interpretation? This is Gene Fluidics. It's a UCLA spinoff that we're doing some work with on some grants. This is the way genetic testing has been, personnel intensive, big, clunky machines. This is what they hope to get to, a 4 by 4 by 4 inch cube. They're not going for the home market yet. But if somebody else figures out what it is, I bet they would jump in there. They're using a biofluidic chip and a MEMS, a microelectromechanical system device, a DNA microsensor array. They're using it now for biological agents, and I've been interested in the rapid diagnosis of infectious disease throughout my career. We've done some work in the past on using DNA for rapid diagnosis of infectious disease. Now the grants that I write with Chi-Ming Ho, the founder of this company, and with the company, it all has to do with personnel portable devices. So you can imagine what kind of money we're going for. If you can diagnose anthrax, you can diagnose bacteria in babies. Too, and the money's in anthrax right now. So they're going for, right now, they have to use equipment that's hauled around by a Humvee to try and pick up these agents in air. And we're going for, or Gene Fluidics is going for, a device that you'd be able to clip onto a service person's uniform. But it can then do other things. If it's DNA is DNA, so it'll be able to do genetic diseases as well, and proteins. We started talking last year, and we started thinking about when a genome costs $1,000, and we weren't sure when that was going to be. I'll show you some companies, I think maybe actually down here, have this as part of their business plan. What is the problems with acquisition? Well, there's going to be an error rate. 3.2 billion base pairs, you can have a pretty low error rate, and you're still going to see lots of errors when you have that size database. What about the interpretation? The analogy here is that it's like whole body scanning. You're going to find differences, and you're not going to know whether those differences really mean anything or not. So there's going to be a lack of an evidence base, and this is why I was saying in genomic medicine we're going to have to enroll whole populations, very large groups, into these studies to give us the evidence base, because otherwise we're not really going to know what to do with this information. What about privacy and information storage? How vulnerable will that be? And then discrimination. It's not only discrimination like genetic discrimination, it's the ability to discriminate between, at a medical level, decision-making level, between what's abnormal and what's normal. But then we know, and we know historically, we made this mistake with sickle cell carrier screening, that we are going to end up mistaking variation for abnormality. So this is from the scientists back in October 14th of 2002, and later on this is, it was Solexa, I think is the group that is working on this. And later, a couple lines down from here, it said that they expected that they'd be able to sequence a genome for $1,000 in 3 to 5 years. Another vignette, a family has a 5-year-old who has a rare genetic disease, has been followed in the same clinic since she was diagnosed in the neonatal period. They move to a new state and are told that the care will be altered significantly because this new clinic has a different approach to management. And as a clinical geneticist, I can tell you they're frequently upset, frequently concerned, and don't understand why these changes are made. But right now, different clinics, different physicians have different approaches. And this is because we consider it an art, not a science. And I'll have my own interpretation of the evidence base compared to other geneticists. The decisions are practitioner-based. They're not subject to regulation. The art of medicine is not subject to regulation. The AMA would fight that one to the death because it's so important to the way medicine is practiced right now. The case series for most rare genetic diseases are quite small. I'll have 2 cases of this in my clinic and write a paper, and Michael Kabak will have 3 cases down here, and he'll write a paper, and pretty soon the world's literature is based on 6 cases done 3 different ways. Of course, the way we do it is right. But that's the way decision making is done. It's not organized for iterative improvement. So I think that we need a collaborative, multi-institutional, protocol-driven clinical studies in genetics if we're going to develop an evidence base when individual patients are rare. An example from genetic diseases, the double-blinded study of the value of penicillin prophylaxis for sickle cell disease. The study was terminated early because the kids on placebo were dying and the kids on penicillin prophylaxis were not. And it is the basis for newborn screening in this country for sickle cell disease now. It was a large protocol-driven study. The classic one is Children's Oncology Group, COG. At least 85% of children with cancer are enrolled in research protocols. Actually, 85% are enrolled in treatment protocols. 95% of all kids with cancer are enrolled in some sort of protocol, be it treatment or natural history. It's done with people, committees, evaluating the data, making an iterative change in the intervention, and it's credited with success of pediatric cancer outcomes that we've achieved, that the field has achieved. The other thing is that I'll argue that even for common complex diseases, the individual genotypes are going to be rare, so we can use the same approach. If you think that you have 25 genes involved in diabetes, let's say, just out of the air, and for any individual, there will usually be 4 or 5 of those genes, and there are going to be multiple sequence variations in any one of them. When you put all that together, the individual genotypes are going to be quite rare. So we're going to have to accumulate large populations. It also means we can approach them similarly as we can for rare diseases. So the highly individualistic approach that provides medical geneticists with management autonomy delays the progress unnecessarily. And physicians are heavily into this autonomy thing. I think we need an organized national approach, and I think it's going to require a buy-in by the medical genetics community and resources to support the protocols. There's going to have to be money that flows for the infrastructure. You're asked to enroll in a genetic predisposition study. It will include those who agree to participate from at least 500,000 individuals who have been selected randomly. Participants will answer a detailed questionnaire and will have mutation analysis performed for the gene of interest. Participants will be followed throughout their lives to determine whether or not they develop this or any related diseases. So some questions you're going to have to ask. Population, is it representative of the general population? Is it biased in any way? And that could be by self-selection. There could be biases. If I've got a family history of cancer, maybe I'm going to be more likely to enroll in this study. Data release, is it going to be released to the participants? Is it going to be released to their healthcare providers or to insurance providers? Who's paying? That's going to determine a lot about this. And is the medical care going to be paid by the study? What about the uniformity for diagnostic criteria? Are practitioners going to be trained so that there'll be better uniformity? And what about uniformity and ongoing data collection? An example of a large clinical study is the hemochromatosis study going on right now. It's about 100,000 individuals to study the relationship between genotype and phenotype for a disease that has a very low penetrance, meaning that many people with the genetic predisposition don't develop the phenotype. Cystic fibrosis newborn screening studies were implemented in Colorado and Wisconsin. Colorado used historical controls because the IRB felt that they could not use blinded controls. But that's been a problem for interpreting the data because the prognosis for that disease has been improving. So you're not sure how you can compare to historical controls. And in Wisconsin, they did blinded controls. And there are issues with that as well. They did have one suit because they kept the data on half the kids, the data were not revealed for 4 years to see how those kids would do compared to the others. Learned some very interesting things. Learned some problems about early diagnosis that really influenced the entire CF community. The kids who were early diagnosed and early started on therapy developed colonization with bacteria because they went to clinic. And it turns out we aren't real good at infection control among toddlers in clinics. So we've got to do better with that. It doesn't mean that screening was bad. It means that we have bad infection control. California is going through a tandem mass spectrometry pilot study at this time. real issue because every hospital had to have IRB review, and it held up the study and still isn't rolled out in all the hospitals. The vignette was based on some of these biobanks. The classic one is the UK Biobank, and there was an article in the New York Times December 31st on this. Planning was begun in 1999. It won't begin until this year because of a lot of ethical discussions. The goal is over the next 10 to 20 years to investigate the role of genes in environment and common diseases. It's planned in anticipation of time when individuals will be able to sequence their genomes, will want to know the implications for their disease risk. It's going to cost $120 million. It's financed by the UK government, the Wellcome Trust, and the MRC. It involves 1.2 million healthy individuals ages 45 to 69. They'll give blood specimens. DNA will be prepared and frozen. 500,000 will be chosen from that group and followed for 10 years through National Health Service records. At intake, they'll have a 10-page comprehensive questionnaire. I mean, I hate the 2-page ones that I do when I go to the doctors, and I'm not sure I'm terribly accurate on those, but this will be a 10-page one. 10-day diet diary and a brief health exam. The data will be held separate from the National Health Service record, but people are concerned about the firewalls there. It's anonymous, but not completely, because people can go in and change the information. So presumably they can read it if they can change it. And these will be their physicians and information from new questionnaires. They anticipate by 2014 that over 40,000 will have cancer, diabetes, heart disease, or stroke. 6,200 will have dementia, hip fractures, Parkinson's disease, or rheumatoid arthritis. They're using SNPs, single nucleotide polymorphisms. They'll compare SNPs with epidemiologic information. And Milburn, who's the UK Health Secretary, has called this a flagship project on molecular epidemiology for the new century. But because of the ethical concerns, it hasn't started, and critics don't share his opinion. They feel that individuals will be exploited, their privacy will be invaded, there will be access to these data by courts and pharmaceutical companies, and that there are future studies that may be permitted and are not fully defined. And yet, if we don't do this sort of thing, we won't have an evidence base. Just a history of these biobanks. The Nurses' Health Study was begun in the 1970s with over 120,000 samples. 1980s saw an 80,000 biobank in Sweden. The Framingham study, which is small, the American Cancer Society started one. 1992, EPIC brought together 10 European countries, 520,000 samples. The Marshfield Clinic, all these had names. I like this one, Personalized Medicine Project, which is another name for genomic medicine. Marshfield, Wisconsin, they have 42,000, and between this year and 2005, 7 projects are planned with sample size between 50,000 and 1 million. This is the last one, but it's very timely. On December 27th, 2002, at a press conference at a Holiday Inn in Hollywood, Florida, the birth of a healthy 7-pound baby girl was announced. She was born the day before. She was nicknamed Eve. She was born at an undiagnosed location outside the United States to a 31-year-old American woman from whom Eve was cloned. I put in the American because just it'll be interesting when she goes for citizenship, I think, with these claims and the feeling of our government about this. The announcement of the birth of the first cloned human was made by Dr. Brigitte Bosselier, a chemist and the head of a private company called Clonaid. She said that 4 other women were pregnant with Clonaid-created clones, and 20 more women were scheduled for implantation of cloned fetuses in January 2003. Clonaid was founded by a former race car driver who also founded the Rallian religion, which he describes as a worldwide atheistic religion of 55,000 followers. They spoke at the cloning meeting at the IOM. There were 3 groups that said they were in the process of developing cloned humans, and this was one of them. And they said there that they believe that we all are descendants of cloned aliens brought to the Earth in 25,000 years ago. So and I think this is important for the science because they consider all of us clones. And I think there could be some issues about whether they might consider normal fertilization cloning. Though they claim to be cloning, they really consider all of us to have derived from clones, and that may be part of the issue. They also, it's central to their religion is the feeling that memories and consciousness transfer from an individual to their clone, and therefore cloning is their path to eternal life. I mentioned two other groups have said they are close to creating human clones, one in Italy and one in Kentucky. The Italian government has said that the Italian physician involved in this will lose his license to practice medicine if they learn that he has cloned, in Italy, if they learn he's cloned a human. No other primate has been cloned, and there are a lot of problems with the cloned animals. The large offspring syndrome, there seem to be some problems with imprinting, some unknown problems with a number of different mammals that have been cloned developing overwhelming pulmonary disease and dying after birth. And that's not understood at all. So there are going to be health risks for the clones and possibly for the mothers as well with the large offspring syndrome. The IOM committee recommended that reproductive cloning should be banned with actually criminal penalties because of the risks to the mother and the fetus. That hasn't happened. Therapeutic cloning should be encouraged. And the issue of cloning and the data from other mammals should be reevaluated in 3 to 5 years. So it wasn't that it's a fundamental ethical issue, but it was some unsafe medical practice at this time, and we needed to get more data. So in summary then, genomic medicine offers great promise. Perhaps not as great as all the geneticists are saying, but still terrific promise. But there will be significant challenges to the implementation of this. We're going to have to see significant changes on the part of the practitioners and the public that we talked about before, and it will foster significant tension between the art and medicine of science. I think that I'm a medical geneticist, so obviously I think we're going to have a role. And as I was putting this talk together, we've been trying to figure out what our role is going to be, and I was thinking about pattern recognition and all of this data, and the fact, and genetics is going to penetrate throughout all of medicine, but how are we still going to, what's going to be the role of a geneticist when it's everywhere? And I thought of the analogy with radiology and imaging technologies. It's used by all areas of medicine. We see the data, we look at the data, and I still miss things when I look at those images. We need specialists to interpret that for us and to help develop the technology. And I think that's going to be the role of geneticists. We're going to be the radiologists of genomic medicine, the individuals who see the fine points and not just the larger, grosser picture. So we've come a long way. The Human Genome Project is really a remarkable feat. The concept that 50 years after we learn the structure of DNA, that we will know the sequence of nearly all of our genome, and how that's changed the way we do research, the way we, at least beginning to change the way we're practicing medicine, is really phenomenal. But also, we have to address these ethical, legal, and social issues. I think it's so important that you're doing that here because, as Al Carnesale said, we probably won't get it completely right this time, but we need to do a little better job than we've done in the past. Thank you very much. So Dr. Kabak is asking about use of all of this genetic information, huge amounts of genetic information for reproductive decision making. Just to give an example, I know you know this example, but I think it's an important one because it says that people are already using this information. In New York City, among a population of very conservative, I think they're Hasidic, very conservative Jews, they still have arranged marriages. They also do not believe in abortion. And so it was recognized that they could prevent this being an issue by including, along with the classical information that they used in mate selection for arranged marriages, now screening for carriers for the diseases that have increased frequency among the Ashkenazim. And so they now, as part of in high school, in this community, they test the boys and the girls. They don't disclose to them what they're carriers for, but they provide that information to the person who sets up the coupling. When I first heard about this, I was on a review panel, a grants review panel, and I first heard about this, and I thought, oh my God, this is really a horrible use of the technology. But as I thought about it more and more, people use different information. You know, it's better information than most of us used in mate selection. And if this is part of their culture, and this prevents the horrors of Tay-Sachs disease, and prevents them being faced with decisions that could really impact tremendously on their faith regarding abortion, I decided, my own personal decision, that this has been a decision made by the community. People know they're being tested. They know how the information is going to be used, and it should be a community decision. But people are very creative in how they use this information. Now, for pre-implantation diagnosis, this is something that we got into in the Secretary's Advisory Committee on Genetic Testing, not pre-implantation per se. But you can take, in a newly fertilized egg, it's done in vitro fertilization, get multiple embryos. They develop to the 8 to 16 cell stage. All of the cells are completely totipotent, can become anything. And that's good because frequently cells die at that stage, and it's good that, if one of your 16 cells die, you still have an arm. 2 arms, so that nature was wise in doing this. But it also means that now with technology, you can suck out one of those nuclei. You can PCR amplify and look for mutations. So where there's a positive family history, you can do this and select. And that's been done. Last I heard, 60-some diseases the testing was available for with pre-implantation diagnosis. What Michael's driving us to think about is the concern that we don't do newborn screening for adult onset diseases. The American Academy of Pediatrics has strongly taken the position that we should not screen for diseases like breast cancer. We could, when we could screen for that more than the 5% of the population we now can, you could identify that disease. Some other diseases that you're not going to change how the person approaches their life in the first 18 to 21 years. Let them make the decision whether they want this testing. But now, what Michael's driving us to think about is saying, okay, it's the same issue, an advanced disease, somebody who could have been very productive, but the parent makes the decision that they will do pre-implantation diagnosis and not have the possibility of this adult onset disease.
Or abortion.
Yeah.
Which raises a different ethical question.
Right. Or prenatal diagnosis and abortion. I think the issue is, and it allows me a chance to come back and address a point in the previous question that I meant to comment on, the fact that if you look, 5 to 10% of the genetics really plays a role in 5 to 10% of disease. I think the problem is that with these common complex diseases, there's low penetrance, and it's what gets you first. And some of it's going to be toxicogenomics or pharmacogenomics that we don't understand. My guess is it's going to be more than that. But what we're going to find, if we really had a complete knowledge of genomics, which I don't think is going to happen in my lifetime, but if we really had that complete knowledge of genomics, I think it's going to be hard to choose because there's not going to be one of those embryos that you could really say is free of disease or free of the predisposition to disease, that you're going to find that they're all. So, you're going to say, okay, I'll take the one with Alzheimer's, but not the one with breast cancer. I don't know how we're going to make those decisions, but I don't think it's going to be, right now we can say, this embryo does not have Duchenne will not have Duchenne muscular dystrophy. This one will. I think when we get to these common complex diseases, we're going to find that we can't discriminate. So the issue then is having a lot of information on adults and them using this information for pregnancy termination. I think that's an issue. And in the Secretary's Advisory Committee, we began to try and look at genetic features also. I mean, then are you going to do it for blue eyes but not? What decision, where's the threshold for this? And I think that's why we need to think about these things and talk about them. And I'm pleased, that's why I put up the slide. There really are a lot of venues for beginning to do this, including the one you have here. I think it's very important that we talk about these and try and figure out how to deal with that, because are we going to say, it's very hard to decide, there's no bright line between disease and trait, non-disease gene. As we get into behavioral genetics, this is really going to open up a lot of issues. So I don't have an answer for that, but I think it's one of the reasons why we have to have open discussion of these things.